In this video, we will discuss about how this DiaBZl stimulates STING pathways in this high affinity engineered T cells. In the previous video, we have understood that DiaBZl will cause increase in the cytotoxicity of the engineered T cells against the tumor cells. So how these things happens? One of the hypotheses is that these engineered T cells will stimulate STING signaling pathways when they get treated with DiaBZl, and the concentrations that we have find out is around this 1 microgram of the DiaBZl is the optimum concentrations at which cytotoxicity will be maximum of the engineered T cells against the tumor. So that's about the previous video. In this video we will understand how they check their hypothesis that DiaBZl stimulates STING pathways in this high affinity engineered T cells. Since high affinity engineered T cells has more cytotoxicity against the tumor cells, so they check on the high affinity T cells, and what they have find out is that after 3 hours they can see several components of the STING signaling pathways gets activated, and they have checked their presence with the phosphorylation of these different proteins, such as this phosphorylation of this TBK1, phosphorylation of this IRFK3, which results in the increase in the pro-inflammatory cytokines, and phosphorylation of this P65, phosphorylation of this STING, and this actin is the control. So what they have find out is that after 3 hours there is a phosphorylation of these components of the STING signaling pathways, and these are higher, or some of these phosphorylated component of the STING signaling pathways are not present in the control where it is not treated with DiaBZl. And these things, some of these things they have quantified also in the panel B, where we can see that the phosphorylation of this P65 in the control case is statistically or significantly lower compared to the case where DiaBZl treatment occurs for 3 hours. And same results they have observed for this PIRF3, which is responsible for pro-inflammatory cytokines, and this P-STING, phosphorylation of this STING pathways, is also statistically significant. And this phosphorylation of this TBK1 is also higher in the case of in the case where DiaBZl treatment is done for 3 hours, and compared to the case where there is no DiaBZl treatment of the HATs. Thereafter they check some of the cytokines presence at different time intervals, and they have selected this IFN beta, CXCL10, and this IL6, which is the pro-inflammatory cytokines. And what they find out for this IFN beta, after 3 hours the increase in the IFN beta signals is significantly higher compared to the control case, and for the 20 hours the signal further goes up in the case of where HATs are treated with DiaBZl. And for the case of this CXCL10, what they have find out is that for 3 hours there is an increase in the CXCL10. However, for 20 hours the CXCL10 goes down for the DiaBZl treated conditions. However, its amount is still higher than its control case. And for the pro-inflammatory cytokine, that is interleukin 6, what they find out, the increase in the fold change in the interleukin 6 is higher for the 3 hours, and it is even higher for the 20 hours. So all these results in together indicates that DiaBZl can stimulate STING signaling pathways that will result in enhancement or increase in the pro-inflammatory cytokines or pro-inflammatory factors in a short duration of time. And that we have seen from these 3 hour results after the DiaBZl treatments. And in this Western blot also we can see that several components of the STING signaling pathways gets activated, which can trigger the immune response in a short duration of time, and that results in increase in the cytotoxicity of these engineered T cells against the tumor cells. So I hope you understood this, how they check their hypothesis about this DiaBZl stimulates STING signaling pathways. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.